I first caught the ferns bug in 1968, rather a long time ago. Here I am back then, engaging in tomfoolery with a chap called Nigel Pennick and a rather large male fern. I tried looking for Nigel online. I found only one Nigel Pennick who seems to fit the bill culturally, but does he look likely? Unfortunately, I've been unable to contact him. When I first joined the Fern Society, I was taken under the wing of several mentors, the main one Jimmy Dyce, a Scotsman who generously taught us all he knew. Another important influence was Clive Jermy of the Natural History Museum, but there were many, many others. I haven't got a picture of Andy Hodges, a different sort of influencer, but an important one. During a field course in 1978, he took the mickey out of my interest in ferns, so much so that I decided to take him in hand. I scribbled a field guide to taunt him with, and after the course kept the file shelved along with others until an opportunity came my way. Between 1978 and around 1991, I continued learning about ferns among society members. Then I found myself teaching identification courses for the Field Studies Council and did so for many years at several of their field stations, most often at the Blencathra Centre in the Lake District. Here's an enthusiastic group examining limestone-loving ferns near Sedbergh in Cumbria. The almost but not quite silly field guide I wrote for Dr. Hodges became the basis for a new Field Studies Council guide. It was my first attempt and not very good. Please do not buy the first or second editions. Just see how much you can pay for one if you buy it from Amazon. A great deal of revision resulted in a third edition with which I was at last satisfied. Meanwhile, I was commissioned to produce one of the FSC's folding card guides dealing with the commonest of our British ferns. Around 2018 to 2019, I noticed that the WildGuide series lacked a fern volume. I emailed the executive editor at Princeton University with an almost cheeky suggestion that I could do it for him. He said yes, and in 2020 BRITAIN'S FERNS, etc. came out. I have learnt a great deal from my previous field guide attempts, so this was something to be proud of, verified by a surprise award from the foremost botanical societies in Britain. That annual President's Prize has been won by a constellation of top botanical authors. So I can offer two field guides, both of which work well to my satisfaction. The FSC guide has a conventional dichotomous identification key with mostly line drawings by Mike Hill for illustration, while the WildGuide has a predominantly illustrated key and comprehensive colour illustration of all species. First, we can separate most ferns into two groups with different rhizomes: creeping, in which the fronds emerge at intervals, and upright, in which they form a shuttlecock shape. Only one species, the narrow buckler fern, tends to do both. Next, frond architecture, from undivided narrow and strap-like to highly divided broad and triangular. We will look at these features in more detail later. Also, we may ascertain the form of the sori, the clusters of spore-producing sporangia found usually on the underside of fronds, but not always. If we are to communicate with one another meaningfully, we must know a number of technical terms. We have already mentioned the two types of rhizome, creeping and upright. Ferns are anchored with nutrient-absorbing roots. The new unrolling frond is called a crozier, as is the similar bishop's crook, and the leaves are known as fronds. The frond. A frond consists of a number of named parts, all represented in this quite complicated tripinnate frond. The section of stalk below the leaf blade, usually clothed in scales, is the stipe, and the rest of it is the rachis. The stalk of a side branch is called the costa. The frond blade is divided as follows. The first main segment is called a pinna, its subsegment a pinnule, and if present as here, the third level of division is called a pinnulette. Now let's look at the undersurface. Here we find the sori, singular sorus, the clusters of sporangia where the spores are produced, but with an ordinary camera they are not very well in focus. We can now do better. Thanks to a specialised camera, the Olympus TG6, which happily works underwater, we can take multifocus close-ups with much greater resolution, and with a bit of cropping we can really zoom in and see what's going on. In many, but not all species, the sporangia are protected by a membrane known as an indusium, which shrivels or in other ways will pull away to expose the ripe sporangia for spore release. Here an indusium has dried and shrunk and lifted to expose the sporangia. Remember the pre-shrivelling tucked under margins of these indusia. They become helpful in identification. Here all indusia on a single pinnule have dried and shrivelled at the same time, exposing the sporangia, some of which are beginning to shed their spores, indicated by the red arrows. The behaviour of the sporangia during spore release is fascinating and surprisingly dynamic, easily observed with a cheap USB digital microscope. A warm lamp helps speed up the process. A sporangium consists of a stalk surmounted by a spherical capsule full of spores. A single row of special cells runs around the capsule from the stalk almost all the way round, the annulus. On the opposite side of the capsule there are a couple of thin-walled cells creating a weak spot. The cells of the annulus are thick-walled on their inner edges, but the outer walls are membranous and permeable. Here we follow the sequence of spore dispersal. As the sporangia matures, tension causes the weak spot to weaken. In the warm, dry summer air, water evaporates through the thin outer walls of the annular cells. Shrinkage of the cell contents causes each cell to contract, and since the cells are all joined together, the tension spreads along the entire annulus. That puts so much stress on the sporangium that the weak spot gives way and the sporangium bursts open. The annulus curves right back until the cell contents can take the tension no more. A small gas bubble suddenly appears in all the annular cells together, releasing the tension and snapping the sporangium almost shut while the spores are forcibly ejected and carried away on the breeze. When a sporangium is ready to pop, the whole process takes just a few seconds and may repeat several times, though getting weaker and weaker. Here on the right we see 3 ripening sori and 3 sori with shrunken indusia and their sporangia ready to pop, showing black. On the left we see all sori where the spore release has been completed. Here we see the complete sequence observed in the common male fern Dryopteris filix mass. First unripe sori, then almost ripe and ready sori with, note the spreading margins of the indusia which differentiate this species from the other male ferns on which the margin is tucked under the sporangia. Next, the beginning of spore release from the first fully ripe sporangia and spore release with, top left, what is left after the spores have been shed. The minute spores are now floating on the breeze, while the heavier spent sporangia tend to fall off and sometimes litter lower fronds. Spores are minute, 30 to 50 micrometers in diameter, and many are thick coated with sometimes intricate ornamentation. The spores germinate and the reproductive cycle begins, but that's not described here. Identifying Britain's ferns. Warning: please do not go straight into picture matching, which often leads to misidentifications. You need to look closely at and compare the diagnostic features, and we'll soon learn where you may use shortcuts to speed things along. Like these people, please use the keys. I learnt key construction the hard way, and you will be grateful that I did. While the first edition of my first published key was developing, I had to learn some strict rules insisted upon by the editor Steve Tilling, who at the time knew much better than I did. I learnt by my mistakes that in a dichotomous key, that is when questions are asked one at a time with just 2 options in each, there is no compromise with its basic set of rules. Couplets, that's those pairs of questions, must always be couplets with a few exceptions, just 2 options in each. The questions in couplets must be mutually exclusive, Character states should not be subjective or vague, and the characters used to direct a user must be available, not seasonal, unless included only for confirmation purposes. Here are some examples of bad identification key construction which will hinder the key user. Two options in each couplet presented in sequence. Here we have 5 stacked together where they would be better presented as a succession of pairs. Couplets must essentially contain contradictions. These two character sets are not opposites, whereas these two express differences clearly. Vagueness does not help the user, while precision does. Height must be measurable. Colours can be misinterpreted. Indeed, colour evaluation is dependent upon colour vision, acute or even slightly impaired, and somewhat tells us nothing. Scale density is subjective, but can be used as confirmatory, but only confirmatory. If important characters might not be available, make sure other characters will enable the identification. For instance, the way the indusia behave as spore dispersal approaches. Some indusia are so thick they are unable to shrivel, so they split, while others roll up or shrivel and fall off. Earlier in the year, these extremely useful features are just not available. And this is very important. An identification key might seem to be helpful and authoritative, but if after you've tried your hardest it doesn't work, maybe it's not your fault. You can blame the author. What do we do to identify Britain's 60+ ferns? To begin with, we take 3 features into account and then follow instructions provided by quick location tables. First, we look at growth form according to rhizome type, either creeping and horizontal or compact and upright. Here are some examples. Above, the creeping rhizomes cause fronds to emerge separated from each other, while below, compact rhizomes form rosettes or shuttlecocks of fronds. So, one or the other. Next, we consider the diversity of frond format. To what degree are the fronds divided? If we consider the stalks to count as naught, then we have undivided, divided once, twice, 3 times, etc. We record that number, 0, 1, 2 or 3, which describes frond dividedness. These degrees of dividedness are given names whereby we may concisely communicate with other pteridologists: undivided, pinnate, bipinnate, tripinnate, etc. The names provide the numbers we need for identification from 0 to 3, plus the 3x3 of bracken and the oak ferns. We then decide what shape the fronds have, the outline, which may be narrow to broad to triangular. We assign the letters A to E to frond shapes. Having noted those features and assigned a number and a letter to your frond, consult a Merryweather guide where at the start, either page 19 or page 15, there is a list of a few ferns that are too different to fit into the identification system, including pillwort and the American water fern. If your unknown is neither of those, you turn to either page 28 or page 40, depending on the book. The procedure is the same. Starting there will soon be your routine, well, until you can flip to the right part of the key that follows or the correct species page. Remember, please don't picture match, well, not until you are proficient. From here on, I will be referring only to the WildGuide. Now, the whole procedure. Identifying ferns using the WildGuide. You have already made a mental note of the rhizome format, horizontal and creeping, or upright and shuttlecock shaped. And you have determined the frond's degree of dividedness and outline, assigning a number and a letter respectively. Decide where the number and letter combination fits in the table, which will lead you to the appropriate page in the keys. While you're at it, observe the form of the sori, a feature that will become useful later. So you have worked through the first part of the key and determined which page we will turn to next. For your next identification, return here to page 28. Having arrived at the keys directed by the table, you will be asked questions, each of them illustrated. The character options, in a couple of cases comparison tables, will lead you to an identification. Each species has a pair of pages with full descriptions, and here those observations of saurus type will assist you. If you go wrong, backtrack as best you can, or, with grim determination, start again. You will get used to it. Let's now have a look at some of the commoner ferns, leaving the more difficult buckler and male ferns until later when we reach the section I call Dips into the Difficult. I should explain a term I will use occasionally, pteridophyte, a word you might also adopt if you become interested in ferns and begin studying pteridology. The word pteridophyte consists of two Greek terms, the pterid half meaning wing or, in fact, just fern, phteri, plus phyte meaning plant, therefore wing plant, which I think you'll agree is not unreasonable. Compare with pterodactyl, those extinct flying reptiles whose wings were supported by their finger bones. Pteridophytes and the study of pteridology provide the Fern Society with its name, the British Pteridological Society. The plant division known as the Pteridophyta consists of several subgroups which includes the ferns, the sole subject of this presentation. The Ophioglossids, of which the world has many more species than Britain, the moonwort left and the three adder's tongues, have long been included with the ferns, but they don't look much like ferns. And here's why they aren't, as demonstrated by a group of American molecular geneticists led by Kathleen Pryor. The full reference is in the box, the paper available to us all on the internet. The arrow on the left indicates the progress of pteridophytes from primitive to modern and represents an approximate timeline. These are the modern ferns. And here are the horsetails, which this study has placed as the nearest relatives to the ferns, not those Ophioglossids, which until recently have been considered ferns, indeed throughout the history of fern biology. Right at the bottom of the chart are the most primitive and earliest evolved of the pteridophytes, the lycophytes, these days known as clubmosses, which are not related to mosses. Sorry, the common names were devised when botany was in its infancy. Here we find the whisk ferns, more modern than the clubmosses, but more ancient than the horsetails and ferns. The photograph is of a modern whisk fern, and the inset a possible distant ancestor which lived around 400 million years ago in the Devonian period. According to the findings of the Pryor team, the whisk ferns and the Ophioglossids have a common ancestry well in advance of the ferns. These are the ferns, and these the Ophioglossids. It would seem that calling them ferns has been proved inappropriate, and our initial intuition that adder's tongues are not true ferns has been validated. Thanks to molecular genetics, the evolutionary distance between the Ophioglossids and the horsetails and ferns has become clear. If the unfern-like horsetails are not ferns, then the Ophioglossids, moonwort and adder's tongues certainly are not. One of our most primitive fern families is the filmy ferns, Hymenophyllaceae. The name is an apt description, membrane leaf. These all have fronds just one cell thick and generally require a still, humid atmosphere or they shrivel and die. Their habitats are relatively unusual and specialised. Of the three, Wilson's filmy fern is the most resilient and the commonest. Anywhere you go in Britain, you will find one or more of our three polypodies. We have three because two of them hybridise by a mechanism that results in a third distinct species in a single generation. Polypodium interjectum, the intermediate polypody, is common but more variable than its parental species, so we infer that its generation anew is not a particularly rare event. Thanks to Professor Irene Manton and her students at Leeds University in the 1960s, plus recent work carried out in the USA, the explanation of the diversity within the polypodies is now well known. In Britain we have a triangle of 3 species and 3 sterile hybrids between them. The southern polypody, Polypodium cambricum, is, as far as our knowledge goes back, the original European ancestral species. It has the base 2 sets of chromosomes. Our common polypody, Polypodium vulgare, has proved to be the result long ago of hybridization of 2 northern species, Polypodium sibiricum and Polypodium glycyrrhiza. Both have the base 2 sets of chromosomes, but a division failure in gamete production resulted in doubling, so that our Polypodium vulgare has 4 sets. Our third species, the western or intermediate polypody, Polypodium interjectum, has 6 sets of chromosomes derived by the crossing of the common and southern polypodies. Its being a distinct species requires a bit of explanation. First, though, the 3 hybrids. One of them, between the common and intermediate polypodies, is sterile because it has an odd number of chromosome sets, 5. The next hybrid, between the intermediate and the southern polypodies, is again sterile, though with an even number of chromosome sets, carrying genes which are just too different to allow reproduction. The third hybrid is between the southern and common polypodies. It is also sterile, with an odd number of chromosome sets, 3. However, this hybrid is different from the others. Sometimes an anomaly occurs that doubles the number of chromosomes, rendering the new polypody fertile and capable of reproduction and spread. It is a new species. Each iteration of this new intermediate polypody can hybridize with its parental species, but those hybrids are sterile, for the time being, in evolutionary terms, maintaining the eternal double triangle of polypodium, species and hybrids. The polypodies do not always obey the identities we impose upon them, so can be quite difficult to identify, and microscopic analysis is often needed. The WildGuide provides a chart to make the process at least reasonably practicable. It is worth having a glance at the distribution maps, indeed of any of the ferns, which sometimes helps to eliminate some species depending on where you are. Leaving the polypodies, we will feature a selection of the more distinctive fern species, first of the family Adiantaceae, named after the familiar but not very common maidenhair fern, with parsley fern, which is mostly in the Lake District and North Wales, and the little Jersey fern, which in the UK grows only in the Channel Islands. This last is at its most northerly limit, and it is common in milder climates, here photographed on Madeira. We have 2 bracken species, one ubiquitous, which everybody knows, the other found at a single site in the Cairngorms, maybe in Assynt as well. Our rare bracken is also found in Europe, Scandinavia and Siberia. The Cystopteris family consists of 6 species, just 2 shown here, the lime-loving bladder fern and the woodland oak fern. The British spleenworts have the greatest diversity of frond format, from simple and undivided, the hart's tongue inset left, to triangular and thrice divided, the black spleenwort inset right. Here is the wall of a ruined Scottish church absolutely covered in plants of the maidenhair spleenwort. Spleenworts like to grow among rocks and on walls. We have 3 lady ferns, one ubiquitous, the common lady fern, and 2 high mountain species, rare and becoming rarer, victims of climate change. The Theliptris family includes the lovely beech fern, left, and the sometimes rather comical lemon-scented fern. Gently stroke its fronds and smell your fingers. You might catch a whiff of citrus, or you might equally well scoff that it smells of green. The family also includes the marsh fern, which grows in undisturbed fenland, so it is much rarer than it used to be because of habitat loss. We still tend to refer to the hard fern by its old name, Blechnum. It is very common on heath, moor and in woodland, and often grows side by side with the lemon-scented fern. It has separate specialised fronds for spore production, emerging vertically from ground-hugging rosettes of sterile fronds. You were warned. We must confront the difficult ferns. In this picture, the main subject is a gigantic male fern with a fringe of buckler fern fronds showing along the lower edge. There are three groups within the family Dryopteridaceae that we must try to learn for the very good reason that the majority are found throughout Britain and their ecological contributions are significant. First, the three shield ferns in the genus Polystichum, which once learnt are easy to tell apart. However, there are two groups of what, when we were first starting out as pteridologists in the 1960s, we dismissed as the Dryopteris-like ferns that completely baffled us and we gave up. Fortunately, we passed that stage, but increased understanding has meant that the differences in detail have now become additional difficulties. I'm afraid it's as true in pteridology as in other subjects. The more you know you don't know. The buckler ferns may be tricky, but the male ferns have turned out to be horribly complex, indeed the stuff of pteridological nightmare. But we'll tackle them. But before wading into the difficult ferns, here are the three shield ferns of the genus Polystichum. The holly fern, far right, has once divided pinnate fronds. It is quite rare, growing in fissures in limestone rock and in mountain habitats. The soft and hard shield ferns are bipinnate and in texture as their names suggest. It takes a little work to distinguish them, but we provide graphic aids to enable that. A chart compares the identification features of each. For the really difficult ones, I have produced a YouTube video guide to the male and buckler ferns, so that this presentation provides only an overview, which I think you will find sufficient for the time being. We have 6 buckler ferns, 2 of which may be set aside because of their rarity. Their habitats and distribution provide enormous hints to their identities, the rigid buckler fern growing only on limestone in the Pennines, while the so-called crested or fen buckler fern's name gives away its habitat and location. So that leaves us with only 4 common buckler ferns to contend with, but they sometimes do puzzle us. Identification is enabled, so far as is possible, with a chart comparing diagnostic characters of the 4, though we must be aware of overlapping characteristics, particularly between the broad and northern buckler ferns, which are very alike and often grow together. The easiest, at a glance, is the hay-scented buckler fern, which does not smell of hay. At last we have arrived at the male ferns. When I started out there were 2, perhaps 3 species recognised, and in recent times one of them was divided into 3, making 5. However, recent research has continued to split the male ferns further, so that to date we have the mountain male fern and the common male fern, plus 8 that not so long ago were 3, in what has been called the Affinis complex. It is not unlikely that more will now be discovered, while 3 Affinis species are common and not impossible to distinguish, so I think we'll stick with those for the time being. 5 tricky species is enough. Therefore, the WildGuide identification key presents just 5 male ferns in detail: mountain, common, golden scaly, boris scaly and narrow scaly male ferns. Use the illustrated key for identification and the table of characters for confirmation. And good luck! Note that 2 types of character are provided here, those which may be defined and those which must be used only as a guide. Other books, of course, are recommended, though remember that the science is moving on rapidly, so older books will be out of date. Chris Page's Ferns of Britain and Ireland contains a great deal of biological and ecological information which identification keys must omit, while his new naturalist volume is a must-read. Chris provided vital advice for male fern identifiers to remember. The satisfactory morphological identification of all the British and Irish variants of this taxonomically very complex group of species usually requires a symphony of characters to be taken into simultaneous account. That is, just 1 or 2 characters will not give you an identification. You have to consider everything available. Male fern diversity is caused by a little-known process, apomixis. The mountain and common male ferns produce male and female gametes and reproduce normally. The other 3, indeed the 8, are male only and reproduce vigorously either vegetatively or by hybridization with female gametes of other male ferns which produce them. Male ferns grow throughout the world, so these phenomena can occur much more frequently than in our biodiversity-impoverished little country. Even Darwin and his contemporary biologist colleagues recognised the impacts of apomixis, though they had no idea what was going on.
There is one point connected with individual differences which seems to me extremely perplexing. I refer to those genera which have sometimes been called protean or polymorphic, in which the species present an inordinate amount of variation, and hardly two naturalists can agree which forms to rank as species and which as varieties. We may instance rubus, rosa, hieracium amongst plants,
and now, of course, male ferns. Chris Page's main book is no longer in print, but can be obtained second-hand at a price. Make sure you obtain the second edition. The first, though available, was thoroughly superseded by the second. Searches also pull up Roger Phillips's excellent photographic guide, and of course, the new WildGuide. Somebody wanted to get rid of it. The WildGuide presents users with a wealth of detail comparing critical characters of the 5 main male ferns. Let's look briefly at each of the 5 male ferns, plus a couple of newly discovered species, which I know because they grow near my home or, actually, in my garden. The mountain male fern, Dryopteris oreades, does indeed grow mostly on mountainsides, but it is often found growing down to sea level in mountainous regions. It is usually small when compared with all the other male ferns, but in rich soils, such as in this churchyard, it achieves full stature. Compare pinnule structure of these male ferns in each illustration on the right. The common male fern, Dryopteris filix mas, grows almost everywhere in most habitats, but reaches its best in woodland. The golden scaly male fern, Dryopteris affinis, is often bright yellow in spring, greening as the season progresses, with its frond stalk densely covered with scales that provide its common name. It grows in the open as well as in woodland on a variety of soil types. Borer's scaly male fern, Dryopteris boreri, is a woodland plant with slender, not very scaly stalks, giving it a more graceful appearance than the robust golden male fern. It is often found alongside the common male fern, which it resembles until you compare pinnules. The narrow scaly male fern, Dryopteris cambrensis, indeed has narrow fronds, which tend to be quite upright, often with the pinnae rotated out of the plane of the frond, almost to right angles. The pinnule pair next to the rachis tend to be significantly longer than the next adjacent pair. The more I know about male ferns, the more difficult confident identification of this species becomes. That's the 5. So now let me introduce one of the newly confirmed male fern species, Dryopteris pseudocomplexa, growing at Dunvegan Castle on the Isle of Skye. It stands out as a bit different from the others because of the distance between the pinnules, which have plain, untoothed margins. And here is another of those newly described male ferns, Dryopteris pseudodisjuncta, in the Loch Alsh region inland of Skye. The classic specimen is growing next to a scaly male fern with which we can compare it, pinnules first and then sori, which in the golden split when the sporangia are ripe, whereas in pseudodisjuncta they just roll up into little cones. It has been interesting to observe that when the first frost arrives, Dryopteris pseudodisjuncta is the first to succumb, while the more robust golden scaly male fern stays green for several more weeks before senescing. The insect shows both ferns before the frost hits. Then in spring, pseudodisjuncta is the first to flush, the adjacent golden scaly fern biding its time again for a few weeks. The insect shows that first frost picture. The last of the new male ferns I will cover here is significant because I have long been aware of it, wondering if it was an odd form of golden scaly male fern, but not only has it been declared a separate species, Dryopteris lacunosa, but would you believe it? A male fern specially discovered it growing voluntarily in my garden. Here are its pinnules compared with those of 2 other male ferns. In a little coombe a short distance from my Highland home, at Stromeferry, a visiting group of specialists found 7 out of the 10 male ferns, the latest to be named Dryopteris cluthensis. Evidently where I live is a male ferns hotspot. Well, that's how it used to be. When in April 2025 I made my usual routine visit, it was gone. Utterly destroyed by builders creating a back entry for a new residence. I don't think I need to describe my feelings. I have no doubt you will be relieved when I say that's the last of the male ferns in this presentation. While learning about them, we hatched a name and a handy expedient for the many we simply can't name. We call them WOBs, an anxiety relief acronym for Walk On By. If you take on male ferns for the sake of your sanity, please also adopt this attitude. You don't know, so walk on by. At some point you will find yourself confronted with plants that are neither one species nor the other, that turn out to be hybrids, such as this shield fern hybrid. It is very rare and so far restricted to Scotland, but you might have the sort of luck I once had, adding a record to the distribution map. There are several videos about ferns on the Ochtertyre Academy's YouTube channel, and this will bring the number to 5. Please visit. The British Pteridological Society welcomes new members. I joined in 1968, and it was with established members that I was able to learn about ferns and then get teaching. Filix or Felix? Two ferns have names that people, according to Freudian slippage, get wrong. The common male fern and the common lady fern both contain Filix in their name. It is Filix or Filix, which is Latin for fern. The word Felix, meaning happy or lucky, is often spoken automatically until the difference has been noted. So we often have to correct people about Filix, while also checking that they say atherium and not antherium, another common Freudian slip.